Hello students. So in the last session we have studied about the tubular reabsorption which takes place in PCT. Okay. So we are learning mechanism of urine formation, that is physiology of excretion, mechanical part. Okay. In mechanical part we have completed first process of urine formation. What is that? First step, ultrafiltration. अल्ट्राफिल्ट्रेशन में अल्ट्राफिल्ट्रेट फॉर्म होता है एंड इन दैट अल्ट्राफिल्ट्रेट लार्ज अमाउंट ऑफ प्रोटीन्स आर देयर एंड large amount of nutrients are also present because there is filtration of the ultrafiltrate because of the force, and we have studied what is the net effective pressure for filtration. Net effective filtration pressure बता सकते हो क्या है? It is 10 mmHg. Okay, got it? So ultrafiltrate which is formed will further move in the tubular part of the nephron, and tubular part of the nephron consists of PCT, loop of Henle's and DCT. Likewise 8 uriniferous tubules will join to form common collecting duct. Likewise many such collecting ducts which are there, these will further open in the collecting tubule. Collecting tubule will further open in the duct of Bellini, which we have already studied. Okay, got it? So ultrafiltrate जो form हुआ है ultrafiltration में, that ultrafiltrate will move in the PCT, which is the very first part of the renal tubule of nephron. Okay, अभी हम लोग ने last time सीखा था कि which are the substances which are actually reabsorbed in the peritubular capillaries from PCT. Okay, PCT में maximum reabsorption होता है. क्यों होता है? We have learned that. Try to recollect. Because it is having brush bordered cuboidal epithelial cells as well as mitochondria in large number, as well as it is having glycogen. Glycogen is the storage form of glucose. Okay, so lot of energy is present here in the cells of the PCT, because of which the PCT can absorb maximum amount of nutrients as well as salts and vitamins also. Many ions, salts का जो ions रहता है, that is sodium ion, chloride ion, then calcium, magnesium, these all ions are very much important for our body. So these will be reabsorbed in the interstitial fluid, and from the interstitial fluid it will enter in peritubular capillary. Peritubular capillary में क्या रहेगा? Blood रहेगा. So blood में फिर से again these nutrients will be entering. That's why it is known as reabsorption. So कहाँ से reabsorb हुआ? Tubule में से. कौन से tubule में से? Proximal convoluted tubule. That's why it is known as tubular reabsorption in PCT. Okay, so we have studied which all substances are reabsorbed from the PCT. High threshold substances as well as low threshold substances. High threshold substances कौन कौन से हम लोग ने लर्न किए थे जो कि reabsorb होते हैं PCT से? ग्लूकोज, अमाइनो एसिड्स, हॉर्मोन्स, एस्कॉर्बिक एसिड, सोडियम आयंस, कैल्शियम आयंस, ओके, क्लोराइड आयंस, all these are the high threshold substances sorry, which are very much important for our body. So that's why they are reabsorbed, and for reabsorption ATP molecule is involved in that. So many ATP molecules will be needed for reabsorption of these high threshold substances. Okay, so that's why such type of reabsorption will be active type. So active transportation method से ये high threshold substances लिए जाते हैं. So very important thing which you have to remember here is that glucose is the main source of energy. Okay, so glucose जितना भी रहेगा उतना body को लगता ही रहता है. So body will try to take and store the glucose in the form of glycogen in its cells. ओके, सो जो अल्ट्राफिल्ट्रेट फॉर्म हुआ है उसमें जो ग्लूकोज है दैट ग्लूकोज विल बी रीअब्जॉर्ब्ड. ओके, बट देयर इज सर्टेन लिमिट फॉर द ग्लूकोज कॉन्सेंट्रेशन इन द ब्लड ऑल्सो. ओके, सो नॉर्मल ग्लूकोज कॉन्सेंट्रेशन इन द ब्लड इज हाउ मच? इट इज 100 milligram per 100 ml of blood. Okay, so 100 ml of blood is having 100 milligram of glucose, and threshold value for the concentration of glucose which should be there in the blood, maximum कितना रहना चाहिए 100 milligrams. So it should not exceed 180 milligrams. Okay, so high threshold value should be 180 milligrams. So 180 milligrams तक टॉलरेट कर सकता है ग्लूकोज को ब्लड. उसके आगे अगर ग्लूकोज का कॉन्सेंट्रेशन ब्लड में बढ़ गया तो what happens, now this glucose is enough for our body will try to eliminate this glucose in the urine. Okay, so if the glucose concentration exceeds 180 milligram per 100 ml of blood, then this glucose will be excreted out in the urine, and such type of disorder is known as glucoseuria. Glucoseuria or glycosuria. Okay, one and the same. Glyco is also word for glucose. Please don't get confused with glucose and glycogen here. Okay, so glycosuria and glucoseuria means what? Excess of glucose present in the urine. So excess glucose जो blood में होता है, that excess glucose has to be thrown out of the body, and that excess glucose, how it will be thrown out of the body? Through urine. Okay, and if such condition arises, then such type of condition is known as glucoseuria or glycosuria. Okay, and this condition is seen in the patients who are suffering from diabetes mellitus. Okay, got it? So very important from exam point of view, what is the disorder if there is excretion of glucose in the urine, and what is the normal blood glucose value and threshold value. Okay, threshold value कितना है 180 मिलीग्राम, मैक्सिमम इतना रहना चाहिए, उससे ज़्यादा हो गया तो excrete करेगा. Got it? Then one more thing which you have to remember, we have studied tubular reabsorption in PCT. So tubular reabsorption in PCT में water reabsorb होता है. कैसे reabsorb होता है? पहले तो solutes, ये जो high threshold substances है, these will be reabsorbed. So solutes कहाँ जा रहे हैं? Interstitial fluid में से peritubular capillaries में जा रहे हैं. Tubule में से interstitial fluid में गए, interstitial fluid से peritubular capillaries में गए. तो when the solute are entering in the peritubular capillaries, along with that water is also entering. Okay, so water को जाना ही पड़ता है solute के साथ, इसीलिए उसको बोला है obligatory reabsorption of water. Okay, very important. So water reabsorption कैसे होता है PCT में? Obligatory. Water is obliged to follow the solutes. Okay, got it? Now we'll talk about the tubular secretion in PCT. I hope everyone knows what is tubular secretion. Tubular secretion का मतलब here addition of the substances from the blood in the tubule. So कौन से वाले substances add होंगे nephric tubule में, that is PCT में, from the blood, which are not important for our body. Okay, which have become excess, those will also be eliminated in the tubule. Okay, so that's why it is known as tubular secretion. और कहाँ पे लर्न करें हम लोग? PCT में. So let's study tubular secretion in PCT. So look at this diagram. What is this? This is PCT. PCT, proximal convoluted tubule, and this is the brush bordered epithelium of PCT. Okay, and this gap between this PCT and peritubular capillary, what is that? It is interstitium. Okay, which is having interstitial fluid. PCT कहाँ पे रहता है? किडनी में, रीनल कॉर्टेक्स में. सो रीनल कॉर्टेक्स में जो इंटरस्टिशियल फ्लूइड है उसमें पहले क्या हो जाएगा, these ions, unwanted substances which are present in the peritubular capillaries will firstly enter in the interstitial fluid and then slowly and gradually they will enter in the PCT. Okay, got it? So कौन से वाले substances हैं? pH मेंटेन होना चाहिए ब्लड का, अगर एक्सेस H पॉजिटिव आयंस हो गए तो pH में changes होगा. So this whatever H positive ions which have become excess will enter in the interstitial fluid and then they will enter in the PCT. Thus H positive ions will be secreted out. Similar way, ammonia is quite toxic for our body. So ammonia will be excreted out in the form of ammonium ions. Ammonium ions will enter in the interstitial fluid, from interstitial fluid in the PCT. Then K positive ions and drugs. We take some of the medications are quite harmful for our body. Only less quantity of that is useful and rest all is the waste. So it has to be thrown out. So drugs and many pigments. Many medicines do have different pigments in them. So these pigments are also quite toxic for our body. So these pigments and drugs will also be secreted out in the PCT. Okay, got it? But for secretion also energy is needed, that is ATP. So by active transport there will be release of H positive, ammonium ions, K positive ions, drugs and pigments in the PCT. Okay, so this is how tubular secretion takes place in PCT. And one more nitrogenous waste we have already learned in mammals, hippuric acid is also formed. So this hippuric acid is also waste, so it will be going in the PCT, it is also secreted out. Got it? So now we will proceed further to the tubular secretion as well as tubular reabsorption, which is taking place in the next part of the tubule. And what is the next part of the tubule after PCT? Guess what must be that? PCT is what? Proximal convoluted tubule. उसको proximal क्यों बोला है? Because Malpighian body के पहले है coiled structure. Okay, then this coiled structure is extending in a straight way. Okay, straight tube like way. So look here, this is the straight tubule of the nephron and it is taking U-turn. Okay, so how is this? Next part of the nephron, it is U shape having two parallel straight tubes. Okay, got it? So this tube is turning in this way. So U shape loop of Henle is the next part of the nephron. Okay, so in the U shape loop of Henle, what will be reabsorbed and what will be secreted? Actually speaking, loop of Henle is the region wherein there will be minimum tubular reabsorption, okay, and tubular secretion. Got it? Why? Because it is more involved in osmoregulation. Osmoregulation means water and salt balance वो maintain करता है interstitial fluid में. Okay, so that's why what is the significance of loop of Henle? To maintain high osmolarity of interstitium of medulla. Okay, किडनी का मेड्यूला है उसमें जो इंटरस्टिशियल फ्लूइड है वो हमेशा हाइपरटोनिक रहता है. ओके, बट टू maintain the osmolarity there is intake of water and intake of salts depending upon the concentration, okay, of the filtrate. So high osmolarity हमेशा maintain रखने के लिए मेडुला का loop of Henle's रहता है. Okay, so loop of Henle's is having minimum role in tubular reabsorption. Okay, got it? So let's study about this. I have drawn the loop of Henle. This is the descending limb and this is the ascending limb. Okay, descending limb of loop of Henle is permeable to water, but it is impermeable to salts. Okay, wherein क्या होता है, वाटर ट्यूब्यूल में से जाता है, मीन्स अल्ट्रा फिल्ट्रेट में से वाटर गया तो सॉल्ट का कॉन्सेंट्रेशन फिल्ट्रेट में बढ़ेगा. नेफ्रिक फिल्ट्रेट जो डिसेंडिंग लिम्ब में है, इट विल बिकम मोर कॉन्सेंट्रेटेड एज इट विल मूव इन डाउनवर्ड डायरेक्शन. पहले कॉन्सेंट्रेशन कितना था, पीसीटी में से जब ये आ रहा था तभी पीसीटी में जो नेफ्रिक फिल्ट्रेट रहता है, इट इज़ of similar osmolarity as that of the blood plasma. Blood plasma का osmolarity कितना है, 300 milliosmol. Okay, so nephric filtrate which is present in the PCT is also having same osmolarity, that is 300 milliosmol. So 300 milliosmol का जो nephric filtrate PCT में है वो कहाँ जाएगा, descending limb of loop of Henle. And when this enters, nephric filtrate of 300 milliosmol is entering in the descending limb, what will happen, as the nephric filtrate is moving in downward direction in the descending limb, water which is present in the nephric filtrate is getting reabsorbed. Okay, so water कहाँ पे गया, interstitial fluid में गया. Okay, मीन्स मेड्यूलरी इंटरस्टिशियम में एंटर हुआ. सो मेड्यूलरी इंटरस्टिशियम यहाँ पे क्या हो गया, डाइल्यूट हो गया. सो यहाँ से वाटर जाएगा, इधर से वाटर जाएगा, सो मेड्यूलरी इंटरस्टिशियम विल बिकम डाइल्यूट. एंड द यूरिन और एल्स फिल्ट्रेट, आई एम सॉरी नॉट यूरिन, फिल्ट्रेट विच इज पासिंग इन द डिसेंडिंग लिम्ब विल स्लोली एंड ग्रेजुअली बिकम हाइपरटोनिक. बिकॉज वाटर जा रहा है तो नेफ्रिक फिल्ट्रेट का कॉन्सेंट्रेशन बढ़ेगा, सॉल्ट का कॉन्सेंट्रेशन बढ़ेगा. सो सॉल्ट का कॉन्सेंट्रेशन बढ़ा मीन्स व्हाट विल बी द टोनिसिटी ऑफ द नेफ्रिक फिल्ट्रेट, इट विल बी हाइपरटोनिक. सो फिल्ट्रेट विल बिकम हाइपरटोनिक. एंड व्हेन द नेफ्रिक फिल्ट्रेट एंटर्स इन द यू शेप टर्न, सी descending limb of loop of Henle is taking U turn. So here in this turn, what is going to happen? There will be maximum reabsorption of water. So concentration of the nephric filtrate in this region will be more. सबसे ज़्यादा concentrated रहेगा. कितना सबसे ज़्यादा concentrated? How much it is? 1200 milliosmol. So पहले 300 था जब ये enter करता है nephric filtrate डिसेंडिंग लिम्ब में, बाद में 600, 400 होता है, 600 होता है, 900 होता है, एंड देन इट कम्स टू 1000 एंड 1200. सो मोस्ट हाइपरटोनिक नेफ्रिक फिल्ट्रेट इज सीन इन द यू शेप टर्न ऑफ लूप ऑफ Henle's. Okay, now whatever hypertonic nephric filtrate which is there, it is now entering in the ascending limb of loop of Henle. Ascending limb is impermeable to water but permeable to salts. Salts especially we are talking about NaCl salt. Okay, so NaCl salt will be reabsorbed, and where this NaCl is entering, it is entering in the medullary interstitium. So medullary interstitium में hypertonic condition हो गया. So hypertonic condition एकदम भी हाइपरटोनिक नहीं रहना चाहिए, इसीलिए वाटर इज थ्रोन आउट फ्रॉम द डिसेंडिंग लिम्ब. वाटर इज रीअब्सॉर्ब्ड इन द मेडुलरी इंटरस्टिशियम टू मेंटेन द ऑस्मोलैरिटी, टू मेंटेन द pH. Okay, pH and balance, ionic balance in the blood, because finally these salts and water, where they are going to enter, in the peritubular capillaries and vasa recta. Got it? Vasa recta रहता है ना उसके आजू बाजू में. So vasa recta is what actually? These are the blood capillaries surrounding the loop of Henle's. Okay, so thus loop of Henle's is trying to maintain the high osmolarity of the medullary interstitium. How it is trying to maintain high osmolarity? Because ascending limb is permeable to salts. So salts जा रहे हैं, किधर जा रहे हैं? मेडुलरी इंटरस्टिशियम में. सो मेडुलरी इंटरस्टिशियम का कॉन्सेंट्रेशन क्या हो गया? बढ़ गया. पहले यहाँ पे क्या हो रहा था, डिसेंडिंग लिम्ब में वाटर वॉज गेटिंग रिलीज्ड, सो डाइल्यूट हो चुका था मेडुलरी इंटरस्टिशियम, सो उसको कॉन्सेंट्रेटेड बनाने के लिए असेंडिंग लिम्ब ने क्या किया, NaCl को रिलीज किया एंड दैट NaCl हैज़ एंटर्ड इन द मेडुलरी इंटरस्टिशियम, दस मेडुलरी इंटरस्टिशियम का कॉन्सेंट्रेशन बढ़ जाता है. ओके, गॉट इट. सो मेन significance of the loop of Henle is to maintain the high osmolarity of medullary interstitium, okay, as well as it maintains water and salt balance in the blood, because whatever water and salts which are released by the descending limb and ascending limb will be exchanged by the vasa recta. Okay, वासा रेक्टा में क्या है, ब्लड है, तो अल्टीमेटली वो ब्लड का पीएच एंड आयोनिक बैलेंस भी मेंटेन रखता है लूप ऑफ हेनलेज. लूप ऑफ हेनलेज में रीअब्जॉर्प्शन मिनिमम होता है, मैक्सिमम रीअब्जॉर्प्शन टेक्स प्लेस इन PCT, that is proximal convoluted tubule. So looking at this diagram, loop of Henle's, you can very well make out the direction of nephric filtrate. Nephric filtrate का direction descending limb में नीचे की तरफ जा रहा है, ascending limb में upward direction जा रहा है. So downward and upward direction, both directions are opposite. So इसको क्या बोलते हैं, काउंटर करंट. ओके, सो काउंटर करंट, करंट जो है फिल्ट्रेट का फ्लो जो है वो अपोजिट है, और ये काउंटर करंट के कारण हाई ऑस्मोलैरिटी ऑफ द मेडुलरी इंटरस्टिशियम इज मेंटेन्ड, एज वेल एज pH and ionic balance is also maintained of the blood which is flowing in the vasa recta. Okay, so vasa recta, where it is present, in which nephron, juxtamedullary nephrons. Okay, so juxtamedullary nephrons are present in the inner side of the medulla. So juxtamedullary nephrons हम लोग ने स्टडी किया था, उसका loop of Henle's बहुत बड़ा रहता है and it is having vasa recta. And what is the main function of juxtamedullary nephron? It is involved in concentrating the यूरिन. सो यूरिन का कॉन्सेंट्रेशन बढ़ाने में वो हेल्प करता है, बिकॉज एक्सचेंज ऑफ द आयंस विल टेक प्लेस बिटवीन द ट्यूब्यूल्स ऑफ द लूप ऑफ हेनले एंड वासा रेक्टा. वासा रेक्टा का लिम्स में एक्सचेंज होता है आयंस का. गॉट इट. सो दिस इज ऑल अबाउट द which all substances are coming out from the tubule of the nephron and which all ions are reabsorbed in the loop of Henle's. So loop of Henle's is trying to maintain the osmolarity of medulla. So this is all about loop of Henle's. Okay. Now in the next lecture we will be learning tubular reabsorption which is taking place in DCT. So tubular reabsorption भी स्टडी करेंगे, साथ में tubular secretion भी स्टडी करेंगे DCT में, so that all the concepts will be clear to you. Okay, till now we have studied mechanism of urine formation में ultrafiltration, tubular reabsorption in PCT, tubular secretion in PCT, then tubular reabsorption in loop of Henle's, and what is secreted out. एक्चुअली स्पीकिंग सिक्रीशन का रोल लूप ऑफ हेनलेज में नहीं है. सिक्रीशन का रोल आपको मेजरली याद रखना है PCT में और DCT में. ओके, गॉट इट. थैंक यू.